Welcome to the Deep Dive of Key to My Research, the science podcast by the Excellence Strategy at the University of Tübingen. My name is Jennifer and I'm your host. In this episode, Hannes Link is joining us. In October 2020, he became Professor for Microbiology at the University of Tübingen. Welcome, Hannes. I'm very much looking forward to talking to you today.
Thank you for the invitation.
Before we get deeper into the area of your research, tell us a bit about your career path. What led you here to the University of Tübingen and make you become Professor for Microbial Biology?
So I took some detour. So I started studying chemical engineering. So it was all about chemistry, how to build chemical plants. And in some lectures, we learned about biology, and I was really fascinated about biology. And I had actually some friends who were studying bioinformatics here in Tübingen. These experiences were really key for my decision to go into microorganisms, study microorganisms, and use them for biotechnological applications. Then I was a postdoc at ETH Zurich, where I learned a little bit the techniques to investigate these bacteria, to use metabolomics, to study how they are functioning and what is actually happening when we engineer these bacteria. And then I was a group leader at the Max Planck in Marburg. This is the Max Planck for microbiology, for terrestrial microbiology. And eventually I became a professor here in Tübingen in 2020.
And when you started to study, had it always been your goal to become a professor and be really deep into research?
Not at all. Actually, when I started my postdoc at ETH Zurich, I had a 2-year fellowship from the DFG to do there my research. I liked doing research, but I also realized it's very competitive. So I thought I'd do this for 2 years and then I go to industry, and that's it with academia. But somehow I stayed, it continued to be a lot of fun. It worked. So yeah, I stayed there. But at no point I decided actively to become a professor.
And what were the key moments in your career path?
I said it before, one key moment was during my studies when I learned about how DNA is translated to RNA, how this is translated to protein, so how the genetic code is read and how this genetic code, somehow, how function emerges from the genetic code. This was really fascinating. I was actually a bit angry that I never learned this at school. So this is a really key moment which motivated me to work on that topic, how DNA encodes all the functions for life.
Mm-hmm. But would it be possible to learn these things at school, do you think?
I think so. Because at the end, it's very simple, right? You have 4 letters, G, T, A, and C, and they make up the genetic code. You translate this, you get a protein, and this has a special function.
So it's not rocket science you're doing?
No. At some point, yes, at some point, no. Yeah.
Okay, we'll get into that in a minute. So Hannes, what is so fascinating to you about microorganisms and especially bacteria? Like, no offense to bacteria, but I rather keep a safe distance from them.
Well, first, they are small. In a way, they're simple. They're simpler than, let's say, an organism like we humans. So I prefer it as a study object because we like to try to study these things at the organism level, at the genome scale. So we really try to understand how the whole system is functioning. For that reason, I like bacteria, especially E. coli, which is arguably one of the best studied organisms that we have on planet Earth. So we study this little bacterium, so it's small. But at the same time, they are very versatile. You can do a lot of things with bacteria. You can produce chemicals with bacteria, so you can use them to address climate change, for example. And on the other side, bacteria can also cause disease, right? You can get infections with pathogenic bacteria, and here also E. coli, it's one of the major pathogens which makes us sick and causes a lot of problems also in the realm of the antibiotic crisis.
Mm-hmm. Just sticking to the topic of bacteria, they're good guys, they're bad guys, but what are their main tasks for our body when they colonize us?
They want to grow, they want to somehow spread their genes, this is what they want to do. And for this, they really developed a wide range of mechanisms, how they can survive in Earth, how they can survive in our gut or even in our nose. So they really colonize special niches, and there they really try to dominate these niches. There's really a fierce competition going on between these bacteria, and I think this is how they evolved. Now, how important are they for us humans? Let's say in our microbiomes, this is something we at the moment try to find out, if they just live there. But from all we know at the moment, we can assume they have a very important function.
Mm-hmm. And what questions do you look at when you're treating the topic of bacteria?
So our main question centers on metabolism. So bacteria, in order to grow, they have to produce building blocks. And out of these building blocks, they produce then all the components that they need to build a new bacterial cell. And bacteria can do something special. This is something we humans cannot do. They can make these building blocks from very simple nutrients. They can take a simple sugar molecule or something even simpler, something like CO2. And out from these simple molecules, they build all the building blocks they need to grow.
Just going one step back, you were talking about the bacterial metabolism. We're not talking about the human metabolism. We're talking about the metabolism of the actual bacteria. How do you define bacterial metabolism?
We focus on bacterial metabolism. So this is all the steps that a bacterial cell needs to reproduce itself. So it starts with a simple molecule. Let's take a glucose, a sugar molecule. And then there are hundreds, thousands of biochemical reactions, and they convert this sugar molecule into the building blocks like DNA, proteins, or the cell wall, which is required to build a new bacterial cell. So think of it as a little factory, for example.
So what is so special about the metabolism of bacteria?
Bacterial metabolism is special and also very interesting to us because bacteria, they have a special property compared to us humans, for example, because they can use very simple nutrients, something like sugars or something even simpler like CO2. And out of these very simple molecules, they can then build more complex molecules, which are then the building blocks for all the components that the cell needs to synthesize a new bacterial cell. So this means in order to grow. Now, sometimes I use the analogy of a factory, like a car factory, but here the factory is building a bacterial cell. So this is the end product. And this factory starts with a simple starting material like a sugar or like something like CO2. And then there are hundreds of individual steps. And in these steps, the cell is building a new bacterial cell as the final end product. And the nice thing is we know each of these individual steps very well. We know what they're doing, where they are in this bacterial factory. But what we don't know is how this is controlled, how all of this functions together.
When you look at these sort of factories, I mean, how can you see them? How do you do your research? How can you sort of understand this process?
This is something we're trying to do. So when I started this during my PhD project, we were able to measure a handful of, let's say, metabolites. These are the building blocks or the intermediate steps in this process of going from a sugar to a bacterial cell. And we could measure a handful. And now we improved these so-called metabolomics methods where we try to measure the components in these factories. So what is made at which time and how much is made. We can also measure the rates of these individual steps. And this has improved a lot in the last years, especially because methods like mass spectrometry are becoming more efficient and more sensitive.
Mm-hmm. And could you just give us an idea of what exactly metabolomics encompasses?
So metabolomics, in the very broad sense, means we're measuring small molecules. This can be the sugar, the direct starting material, or all of the intermediate metabolites that lie between the sugar and the final building blocks, like something like DNA or proteins.
Mm-hmm. And this specific method is used for understanding the metabolism of bacteria.
And with this, we can understand the metabolism. We can see, for example, at some point, is there more of a certain precursor or is there less? Is there a bottleneck in this process of this factory? Because you have to imagine you have hundreds of individual steps and they all have to work together because if one of these individual steps is not functioning, then the final product cannot be made.
Mm-hmm. And can you intercept in this process? Are you capable of like going in and stopping a certain module?
This is the way we try to understand how the factory is working because with techniques like CRISPR, we can now somehow perturb each individual reaction step in this factory and then somehow just see what happens. What is the cell doing if this step is not working or if another step is not working?
Mm-hmm. We're quite deep into the methods right now. Could you just explain what CRISPR is? Because this has been a quite innovative method which has changed a lot for your research.
CRISPR has changed a lot for our research. I think it has changed a lot for everyone in biology. Because with CRISPR, we're now really able to write DNA. So CRISPR, it's called an endonuclease. It's a protein, Cas9, for example. And this protein can cut DNA. And it can cut DNA precisely at the position where I can program it. So it's a programmable scissors. So I can cut the DNA exactly at the point where I like to do this. And at this point, I can then insert a new gene or a mutation, or I can delete something. So it really allows us now to reprogram the genetic code of a cell.
Okay, I have this picture in my head of a real life scissors, but it can't look like this. What does it look like, CRISPR?
Very boring, like a liquid.
Okay.
But inside this liquid, you have then genes that we synthesize. You can buy this from a company. So these are little genes. And on these genes, we can then program where this little protein, which is also in the liquid, then binds to the DNA in our bacteria and where it cuts the DNA and where we can make the genetic edit.
This sounds as if your laboratory must have some kind of special security mechanisms, because, I mean, it's not good if this sort of was open access to many people, right?
Absolutely. So first, I mean, this is genetic engineering. So we have a safety level for genetically modified organisms. So we have to be safe with this. And we also have to take safety measures because we're working with pathogens. For example, we take strains from the clinic, which have developed resistances against antibiotics. So really problematic pathogenic bacteria. And then we can use CRISPR to see now how can we resensitize these bacteria such that the antibiotic is now working again.
How do you keep safe yourself? The laboratory is safe, but what do you do, like clothes-wise, for example?
I mean, we have safety codes in the lab. So whenever I enter the lab, we wear safety codes. When I come home, I wash my clothes. So these are the safety measures that are enough in this case. So we're not working with the very high-risk pathogens like viruses, like Ebola or something. These are safety level 2 pathogens, but we have to be careful.
Okay.
And we are.
Very interesting. So let's turn to a bit broader subject, to synthetic biology and systems biology. What is your focus in this area of research?
So these are very broad research areas. So in systems biology, we try to understand how these bacteria are working. So that we try to understand how are the molecular components inside the bacteria, how they're interacting, and which networks emerge from these interactions. Often this is very messy. These are very complex networks. But these networks really allow the cell to self-organize, to self-repair, and to function in a way how they do. So this is the task of systems biology, that we map out these networks, that we understand how they function, these networks, and then we can use this understanding for synthetic biology. And synthetic biology is really all about genetic engineering, about writing the genetic code or coding with DNA.
And what do you aim at finding out by this second area of research with the synthetic biology?
So synthetic biology has many applications, really a wide range. We all know the mRNA vaccines that we use now for the coronaviruses, but we can also address climate change by reprogramming bacteria, for example, such that they fixate CO2. This is something we do, for example, in a collaboration with the lab of Ron Milo in Israel. So what we do there, we can transplant the CO2 fixation pathway from a plant into a bacterium like E. coli. So E. coli naturally just uses the sugars or something that you can use in our gut. But now we can just transfer a pathway from a plant, let's say, into E. coli and then try to get this functioning such that E. coli is not eating sugars anymore, but that it's eating CO2. And then you can imagine then you can address a lot of problems if you can make chemicals out of CO2.
I didn't quite understand with whom you're collaborating. Could you just come again?
Oh, this is a colleague. He's in Israel and he really pioneered this field of fixating CO2 in bacteria like E. coli. He had some breakthroughs there. He got really systems to functioning. Now the problem is it's working. So E. coli can use CO2 as a carbon source, but it's very, very slow. It's a little bit like you want to build an electric car out of a car with a combustion engine, and it's not enough just to remove the engine from an electric car into a combustion engine or something like this. So we really have to re-engineer a lot of other things, and at the end that you have a functional system which works also with very high rates, for example.
And how much time would this process need to find out all these things?
So this is again the point where CRISPR comes into play. So synthetic biology, I said it's engineering biology, but we're not there yet. So I'm a chemical engineer. So I learned how to build a chemical plant. And this works usually, we design this at the computer. And then we build the plant and we know the plant is working. And this is something we cannot do with a biological system. Let's take the CO2 fixation pathway. In our case, we're not there that we design this at the computer, we install this in E. coli, and it's working. It's still a lot of trial and error. And our CRISPR allows us to do a lot of trial and error, to do this really at a massively large scale. So we're trying at the moment to make hundreds, thousands of genetic changes and then just try which of them is helping E. coli in this case to work with this CO2 fixation pathway. And from this trial and error, we can then kind of iterate a little bit and get some more understanding and then do this in a more rational way.
This sounds like you need to be a very patient person to do this kind of research. Are you?
Oh, actually not. I'm not patient, so we can do this very fast and in very high throughput. That's why we're developing, actually, I think it's a little bit my impatience that we develop high throughput methods so we can construct, for example, these mutants with hundreds or thousands of mutations. We can do this in a few days. So you all do this in one pot. And then you also examine their fitness in a competition experiment where they grow all together and we get fast results.
Okay. So another area of your research is the microbiome, but not any microbiome, a specific one. Could you tell us a bit about this subject?
Yeah, we started now with the nasal microbiome. So in Tübingen, I have many colleagues working on microbiomes. We have Lisa Meyer, she works on the gut microbiome, and this is all very interesting. But since we're working with E. coli and already know how complex the system, a single bacterium can be, it was a bit daunting to me. Now suddenly you don't have a single bacterial factory, but you have many of them and they're interacting. That's why we decided for the nasal microbiome. So it's a little smaller microbiome where not so many bacteria are living together. So we started now analyzing the nasal microbiome. And again, we're focusing on metabolism. We're asking what happens now if a bacterium doesn't work in isolation. So this metabolic factory is not alone. It has now 10 other bacteria. And from all we see at the moment is that they're highly interacting. So these factories kind of work together, they share some nutrients and maybe also develop some new functions.
Mm-hmm. And can you actually actively influence the nasal microbiome? Because, I mean, scholars say, or one reads a lot, that you have influence on the gut microbiome, just how you eat. But what can you do regarding your nose?
This is a very good question because you don't have so much ways to modulate the nasal microbiome. And this is, for example, something we would like to find out. How can we remove a major pathogen from the nose? So 1/3 of humans, they have Staph aureus in the nose. This is this pathogen MRSA, which causes a lot of problems in the clinic. And often if you go to the clinic, you get a cream which you can smear into the nose that Staph aureus is removed from the nose. And there we would like to find out alternative therapies or alternative ways of somehow to modulate the nasal microbiome such that Staph aureus can be removed. And one way could be, for example, probiotics, that you just put another bacterial strain into your nose and then you have a more healthy nasal microbiome.
Mm-hmm. And how come this certain pathogen colonizes your nose? Why specifically this organ? It could go anywhere.
I mean, they colonize a lot of body parts. We have microorganisms on the skin. We have the mouth microbiome, plays an important role in when you go to the dentist and when you have problems with your teeth. So we're investigating antibiotics. So how antibiotics work and how antibiotics kill bacteria, because the antibiotic in the first place, it just inhibits the bacterial growth, so the bacterium cannot grow anymore. But what we actually want is that the bacterium dies, that it really disappears from our body. And there we're investigating a little bit the mechanism that leads to bacterial cell death. And there we learned a little bit that it seems that metabolism plays an important role, because if bacteria find a way to slow down their metabolism, then they can somehow prevent that they die from an antibiotic. And this can be dangerous in the clinic, or if you have an infection, if you get an antibiotic, you take it for several days, the bacteria will not grow, but then actually once you remove the antibiotic from your body, then they can grow again because they're not died. So you really have to clear the bacteria or the pathogens from your body. And this is why we would like to find out, how can we improve this killing? For example, is there a certain diet that you could take with an antibiotic such that the antibiotic is more effective?
Mm-hmm. Or, I mean, broadband antibiotics tend to or do kill all microbes that inhibit the body. But is there sort of maybe a possibility that you can sort of channel it to one certain bacteria? Are you looking at this? Because they have quite a bad image, antibiotics, meanwhile.
Exactly. So this is known. So antibiotics kill basically everything, and this is a problem. So my group itself is not researching on this topic, but there has recently been really a breakthrough from another group in France, and they managed actually to use CRISPR to just target one specific bacterium. So this is nothing we're trying to do at the moment, but it's really interesting to follow that you can now use CRISPR and deliver this genetic scissor to just one bacterium which causes problem to the pathogen in your microbiome and then remove precisely this bacterium.
That sounds very innovative, but let's get back to what you actually do. So what is your specific area when you look at antibiotics?
So we try to understand how metabolism interacts with an antibiotic. The moment we can assume that metabolism plays a role, especially in cell death, and that bacteria that can slow down their metabolism can somehow go around the cell death. So maybe they are not growing, so they're inhibited by the antibiotic, but they're not dying from the antibiotic. And this has a lot to do with metabolism.
And how do you go about when you try to find out these questions?
So here we use again CRISPR. For example, in one project, we just now went into databases of clinical isolates. The genomes are listed in databases. And then we search in these databases for mutations in metabolic genes. Because we can assume maybe there was some pressure on this bacteria that they have to survive a certain antibiotic treatment. Then we look for mutations in metabolic genes. Now, the problem is if you take one clinical isolate, you will find many mutations. So we don't know which of these mutations has some influence. And now we can take thousands of these mutations. We just made a big database out of this. And then we insert each mutation individually, again with CRISPR, into the genome of our laboratory strain. And then we can test if this mutation helps our mutant that we generated to survive the antibiotic treatment or even to grow in the presence of an antibiotic.
So you're involved in so many different fields of research, and they all seem to have, when I listen to you, different aims. Or how do you bring it all together? What is the big goal? Is there one?
So it all starts with metabolism, and that's our main goal. So our main goal is we want to understand how this complex network of biochemical reaction, how this is controlled and how all of this is functioning together. And so really our main efforts is this basic understanding, basic research in metabolism. And if you understand metabolism, then you immediately have a wide range of applications. And this could be microbiomes, where we understand or investigate metabolism in the microbiome, or the metabolism-antibiotic interaction, or synthetic biology, where we try to reprogram metabolism. And our role, often these are collaborations, our role is really to investigate metabolism and to modify metabolism.
So we've just been talking about the real-world applications, but we'd really like to learn more about specific examples. So how does your fundamental research in these areas, how does it translate to real-world, to new therapies? Could you give us some examples on this?
I mean, maybe one example is a collaboration that we have with medical microbiology, with the clinic in Tübingen. So what they often do when you have an infection with a bacterial pathogen, they take isolates of this pathogen, they sequence maybe the DNA of this pathogen. And there we collaborated with the clinic and we just received these pathogens. In one example, this was again E. coli, so E. coli from the clinic. Also strains that developed resistances. And then we started to investigate their antibiotic resistance profiles and their metabolism and try to understand, is there just some connection between their metabolic state and the way how they respond to an antibiotic. And we did this for many strains, but at some point we learned that it's way too complex, because all of these clinical isolates, although it's the same bacterial strain, it's all E. coli, so the same strain, but they differ in their DNA. They have little changes in their DNA, and this can already have a very strong effect on how they're functioning and how they behave. So really none of these strains is equal to another strain. And at some point we really decided to stop and go another way, where we insert now these individual mutations from these strains, again with CRISPR, into our laboratory strain, where we know very well how this strain is functioning. And then we can try to understand the impact of each mutation individually. And with this, we hope to understand a little bit interactions between metabolism and the antibiotic action.
And do the hosts actually know what's happening to their E. coli that they're getting into your laboratory?
I mean, there are a lot of ethic restrictions, right? So we can just take strains where I think the doctors call it general consent. So the patients have to, to say, yes, you can use this bacterium.
For research reasons, yeah. And do you spend any time in hospital at all? Do you have any contact with the doctors or the people on the ground?
Absolutely. So actually the institute we are in, there are many doctors, and this helps a lot to understand what are the real world problems. Are we researching on something which is far off, or is this actually relevant? And this is important to get feedback from the doctors.
And you were just talking about, is this relevant? How do you generate your research questions? Do they come from the practical side? Do you generate them in your laboratory? Is it a big part of what interests you as a scientist, or how does it all come together?
It's maybe the most important part of your research to define your research question. It's also the most difficult one, because, and at one day you wake up and you think you have, the best question ever and you have to study this. And one day later, you realize this was actually a bad idea. So selecting the right research question is important. Usually with my PhD students, we take enough time, but it can also be frustrating if after 1 year you realize this was actually not the right question. And it's just because you learned something on the way, and then you just have to stop something.
And what questions would you like to sort of conquer looking into the future? Is there anything that's very special to you?
This comes still from my PhD studies. So in the future, I would somehow like to be able to have a computer model or a simulation of not a complete bacterial cell. This is very ambitious. Although some of my colleagues in the US, they're trying this. They're trying to simulate a full bacterial cell. But at least what I would like to do, that we have some predictive models of metabolism. So what is it that I have to change to achieve a certain function in a metabolic network, that somehow we can recapitulate metabolism at a computer model.
Mm-hmm. Talking about computer model, maybe about deep learning, what role does AI play in your research at the moment?
We try some things, especially with the colleagues. We have the cluster of excellence for machine learning here, and there are some colleagues that they help us in analyzing the data. Because I said before, with CRISPR, we generate thousands of mutants. In thousands of mutants, we can again measure thousands of metabolites. You can imagine this leads to very large data matrices. And with these data, we're making the first tries to use some data-driven approaches, some AI learning, that we have the big data and somehow predict certain functions just from feeding large amounts of data into a model. And I think the AI will become more and more important because we're generating so much data. We tried a lot to work with mechanistic models, really models which consider each individual part in a cell, and then we put this together and somehow try then to get the same function as in a cell. But I think this is unrealistic because the individual parts in a cell, they're highly connected. They're interacting with many, many other parts. And often, we cannot map the full interactions there. So having mechanistic models seems a bit unrealistic. So I think it's more realistic that in the future, we just have data-driven models that somehow learn these complex interactions just out of the data.
So both your research, but also technology is very fast-paced and evolving. So how can you work in this very dynamic environment? How can you be successful with science?
The key is to focus, and focus especially on the method. So you cannot do everything. So we focused on 2 key technologies, and there we really try to stay up to date. And this is CRISPR genome engineering in bacteria, and metabolomics. And then with these methods, then again, we try to focus on specific questions. In our case, this is metabolism. But from this point on, we can be relatively diverse because we have good methods. They're focused. We have a focus on metabolism. But then, as I said before, there are many applications.
It all sounds very demanding what you're doing. There's probably a lot of pressure in science. And how do you spend your weekends, or how do you spend your free time to get a little balance of this sort of demanding job or profession?
Yeah, it's competitive, and it has a lot of ups and it has a lot of downs, right? Sometimes you win and sometimes you lose. And this is something you have to develop some resilience to this, that, a paper is rejected, but the other day a paper is accepted, and then you can celebrate. I think this is just something you have to learn, that, goes up and down. And in my free time, I spend time with the family. I do some sport. I go running or go kite surfing. So I have other things than science. Yeah.
Other things than science, that's good. Can you really disconnect then when you're out there at the weekend, kite surfing and running?
No.
Okay, the microbiome is always with you.
It stays with you, and I think if you have a particular problem, it stays with you. But I think I learned now over time, you keep the good problems and somehow you get rid of the bad problems. So you kind of incubate the interesting problems. So if you stay with this, I think it's still healthy, and it's just part of the job, yeah.
Okay. And talking about the University of Tübingen, what makes this location so special for you as a scientist?
The Tübingen is great. So when I came here, I was relatively new to the field of infection biology, and we really have some great scientists in infection biology, especially focusing also on bacteria. I learned a lot from these colleagues. I learned a lot of new, interesting research questions. And it's not only infection biology, we also have great scientists in synthetic biology. So again, I can really collaborate on these different research topics. And it seems that everybody or many people are interested in metabolism is also helpful for us.
Not only your direct colleagues, but there must be a lot of sort of science around you that focuses also on the metabolism.
Absolutely. For example, cancer research. So colleagues now in our new center at the clinic, they're focusing on cancer. And obviously cancer is kind of a metabolic disease. They're also interested in metabolism. And they also learned that cancer comes with its own microbiome. So there are also a lot of connections to this research field.
And what makes the work within the cluster special? Or can you tell us a bit how you work with your colleagues, how you work with a cluster?
So in the cluster, the important thing is that we have experts in these different research areas, infection biology, and especially the microbiome. Because when you said it before, it's a very dynamic, it's a very complex and competitive field or fields. So doing this on your own is very demanding. So often these are collaborations where we work, for example, with colleagues on the gut microbiome, and then we can take our methods, work with these colleagues which have other expertise, for example, in these different bacteria. We are focusing a lot on E. coli because, as I said it before, it's simple, it's well known. And then moving already to another bacterium is somewhat difficult, and therefore it needs collaborators. And this helps a lot in Tübingen.
And what other than Tübingen as a scientific or academic location, what makes a city and living here special for you?
It's nice. It has a river. Many young people, in a way, keeps you young to be surrounded by many young people. And it's nicely located here in the Swabian Alps. So it's a nice place.
I'd have one last question, which we ask all our scientists that we invite to the studio. And this is, what is the key to your research?
The key to my research is to stay motivated, that I have questions that I think are interesting enough, that I spend a lot of time thinking about them, and then I can develop, again, interesting ideas. And then maybe the key is just to select the right idea out of these many ideas that you have.
Hannes, thank you for joining us today.
Thank you for having me.
This was Key to My Research, a podcast produced by Changing Time in cooperation with the University of Tübingen. If you enjoyed this episode, please subscribe to the podcast, leave a review, and recommend it to your friends and colleagues. I'm Jennifer, and I'm looking forward to seeing you next time.